Hello everyone. Today we will take a brief idea about DNA replication. I am Dr. Rashijan. Welcome you all. During cell division, each daughter cell get an exact copy of genetic information of the mother cells. This process of copying the DNA is known as DNA replication. So DNA replication is the basis of biological inheritance process by which a double strand DNA molecule is copied to produce two identical DNA molecules. So whenever a cell divides, the two new daughter cells must contain the same genetic material information or DNA like parent cells. So DNA replication is a process like photocopier machine in which you get same print or copy as original. There are three main steps of DNA replication. First one is initiation, second one is elongation, and third is termination of the process. There are three possible replication models. First one is semi-conservative, second one is conservative, and third one is dispersive. In semi-conservative model, two strands of DNA unwind and they both like a template for synthesis a new or complementary strand. The result of this model, two DNA molecule with one original strand and one new DNA strand. So that's why it's called semi-conservative. Semi means half of the DNA is conserved. Second model is conservative model. In this, DNA replication result in one molecule that consists of both DNA strand. It means one strand is new and one strand is old. And third one is dispersive model. As name show, dispersive. So multiple unit are dispersed with old one and new one. This is called disperse. They are disperse each other. So that's model show hybrid DNA. In this model, DNA application result in two DNA molecules that are mixture. So that's why it's called hybrid DNA. This is diagrammatic presentation of three possible replication model. In semi-conservative model, after first replication cycle, one strand comes from parental DNA and one strand after DNA replication. It is one is new and one is original. In conservative model, after first replication cycle, one DNA molecule is same like parental DNA and one DNA molecule is totally new. But in dispersive model, there is a multiple patchwork in both DNA strand. So it is also called hybrid DNA. There are some characteristics of DNA replication. First one is DNA replication is semi-conservative replication. It means one strand of parental DNA is conserved, but second one is new. Second characteristic of DNA replication is DNA replication is bidirectional. At the same time, it turns both sides from right to left or left to right. Third one is semi-continuous replication. And fourth one is highly fidelity replication. Before the starting of DNA replication, we have to know about component of DNA replication. DNA replication would not occur without these enzymes because they catalyze important and various steps in the DNA replication. So they include DNA helicase, DNA primase, DNA polymerase, topoisomerase, exonuclease, DNA ligase, and telomerase. You know, both the DNA strand held together by hydrogen bond between the nucleotides. So DNA helicase break the hydrogen bond between two nucleotides and unwind as well as separate both the strand of DNA. It moves along with DNA strand and responsible for formation of replication fork or DNA bubble. Second enzyme is primase. This is a type of RNA polymerase that generate RNA polymers, which are short RNA molecules and act like a template for starting DNA replication. Next in series, DNA polymerase. This is synthesize new DNA molecules by adding new nucleotide in both the strand leading as well as lagging. Next, topoisomerase, better known as DNA gyrase. This enzyme is responsible for relax the DNA strand during replication. When DNA strand uncoil at the middle, it is always recoil at the terminal end. So it release the tangle or supercoiling at the terminal end. So it unwind and rewind DNA strand to prevent the formation of tangle. Next is exonuclease. So it remove nucleotide from the end of DNA chain. Next one is DNA ligase. Ligase, it means it is act like a glue by forming phosphodiester bond between new nucleotides. And last one is telomerase. This is a special type of DNA polymerase. It is responsible for synthesis of telomer at the end of DNA, and it is responsible for termination of DNA replication. So first step of DNA replication is initiation of replication. So DNA replication start by the breaking of hydrogen bond between these two strands. So both the strands run anti-parallel, 5 to 3 and 3 to 5. So helices introduced in the in the chain, and it break the bonds between the nucleotides and forming the replication fork or DNA bubble. In this diagram, there are some molecular component in the both the strand, leading as well as lagging strand. So the component called SSB or single strand DNA binding protein. So what is the role of SSB? It is responsible for adding DNA helicase and is also responsible for stabilizing the component. When once the hydrogen bond break, both the strand of DNA get separated and gives a structure like a fork, prawns, and now they called replication bubble, replication fork, or replication prawns. One more important thing related with unbonding of two strand of DNA. The splitting happens in place of the chain which are rich in adenine and thymine. That is because there are only two bonds between these two, whereas there are three bond between cytosine and guanine. After forming of replication bubble, second most important step of DNA replication is binding of RNA primase. RNA primase has tendency to bind at the point of 3' to 5' position at parent chain. Primase can attract RNA nucleotide. Collectively, RNA nucleotide called RNA primer. RNA primer about 100 to 200 nucleotide long and is synthesized by RNA primase. So RNA primers are responsible for starting DNA nucleotide because The RNA primer are removed by DNA polymerase by using exonuclease activity and is later on replaced by DNA nucleotide by DNA polymerase. So this is all about step 1 of DNA replication, that is initiation of replication. Now we will move to step 2, that is elongation of DNA replication. Before starting the second step, we will take a short review by animation.
The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA.
The replication is totally different for both the strand. For the 5' to 3' template, the proceeding of daughter strand is continuous because DNA polymerase 3 can read the template continuously in this direction. So DNA polymerase can add nucleotide one by one which are complementary to the parent nucleotide. For example, adenine for thymine. So that's why this strand called leading strand. The process of replication in the leading strand is quickest because it requires very few steps. So once the RNA primer laid on by RNA primase, the DNA polymerase 3 can build the second strand continuously in the same direction that the double helix is being opened. So complete process, DNA polymerase 1 is replaced by DNA primer with DNA by the action of exonuclease. But in case of second strand, this is 3' to 5' template. The DNA polymerase 3 cannot read the template continuously. So the process of replication in this template is not regular and more complicated. So that's why this strand is called lagging strand. In this strand, RNA primase added more RNA primer and DNA polymerase 3 read the template and lengthens the burst. The gap between the RNA primer is called Okazaki fragments. In this lagging strand, DNA polymerase 1 exonuclease read the fragments and remove RNA primer. After removing the RNA primer, the gap left behind. So the gaps are closed with the action of DNA polymerase. This is another DNA polymerase which add more complementary nucleotide to fill the gap. Now DNA ligase which act like a glue to attach the phosphate to the sugar moiety by forming phosphodiester bond.
An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand.
The third and final step of DNA replication is the termination. This process happens when the DNA polymerase reaches to an end of the strand. On the lagging strand, DNA is synthesized in short stretches, each of which is initiated by separate primer. There are 2, 3 or 4. There are multiple set of primer presents. When the replication fork reaches the end of the linear chromosome, there is no place to make a primer for the DNA fragment to copy at the end of chromosome. These ends thus remain unpaired, and over time they may get progressively shorter as cells continue to divide. We can easily understand that when the RNA primer is removed, there is no possible for DNA polymerase to seal the gap because there is no primer. So the end of parent strand where the last primer wind is not replicated. These end of linear chromosome consist of non-coding DNA that contain repeated sequence. They are called telomeres. Telomere is a Greek word. It means end. As a result, a part of telomere is removed in the every cycle of DNA replication, and DNA segment becomes shorter, ultimately processed to the death. So the shortening of telomeric end is prevented by a specific enzyme called telomere terminal transferase or telomerase. Basically, it is a ribonucleoprotein that add a species-dependent telomere repeat sequence to the 3' end of telomere. Telomerase works as a reverse transcriptase enzyme that carries its own RNA molecules, which is used as a template when it elongated telomeres. By the adding nucleotide, it extends the overhanging strand of the telomere DNA using this complementary RNA as template. When the overhang is long enough, a matching strand can be made by the normal DNA replication machinery. That is why using an RNA primer and DNA polymerase, thus producing double strand DNA. While printing a page, the typewriter sets the type and impression is taken. Proofreading is done to correct mistaken if any, and then the final printing is done. A similar follow-up mechanism operates after DNA synthesis. So the replication process should be carried out with high fidelity. Otherwise the information is altered. Hence there should be foolproof mechanism to correct the mistake. So this is a summary of DNA replication. First, helicase break hydrogen bond between two helicals and forming replication fork. Then single strand binding protein coat DNA around the replication fork to prevent rewinding of DNA. Next, topoisomerase work at the region ahead of replication fork to prevent supercoiling. Then primase synthesize RNA primers which are complementary to DNA strand. Next, DNA polymerase 3 extend the primers by adding nucleotide at 3N to make the bulk of new DNA. RNA primer are removed and replaced by DNA polymerase 1, and finally gap filled by DNA ligase. So this is all about DNA replication, and thank you for watching this presentation.